example, they got infected, they got fever. So some cells will secrete shock proteins. So these are also can be recognized by innate immunity, and they start to launch immune response, as well as the altered membrane phospholipids. So we know that PEMS is basically the part of the pathogen that innate immunity recognize. So the next thing we need to know is the match to the PEMS, right? Match to the pattern. Just like when the antibody bind to the antigen, they should have matched, right? So for the PEM, what is actually the matching molecule for them? So in order for the innate immunity to recognize the PEM, they should be expressing a specific molecule or a receptor on the surface of their cells, which is called as pattern recognition receptor, PRRs. So PRRs should be able to recognize PEMs. So where do we find PRRs? Any cells that comprise of PRR should be able to perform innate immunity. So these are the list of cells usually contain PRRs, pattern recognition receptor. Therefore, they have the ability to recognize PEMs. So these are the cells. If you look at one by one, you'll see that these are basically the cells involved in the innate immunity, right? So before this, we just memorize that these cells belong to which type. Now we understand why. Because, for example, macrophages, they have PRRs, and therefore they could recognize PEMs that allow them to be involved or classified under the innate immune response, including dendritic, endothelial, mucosal, lymphocyte, basophil, eosinophil, and so on. And for the pattern recognition receptor, they could be classified into two types. First is for the is known as endocytic pattern recognition, and the second one is known as signaling pattern recognition. So for the first one, it could be a lot of type of receptor, including the mannose scavenger receptor, scavenger receptor, opsonine receptor, and formyl med receptor. So how do we memorize the receptor? It's basically referring to the surface of the PEMs. So this receptor will be bind on the mannose-rich sequence of the bacteria. Scavenger receptor normally a common receptor present on the surface of the phagocytes. So opsonine is basically a receptor that can bind to molecule opsonine. For example, molecule secreted by the complement pathway. And these are also receptors that recognize a specific pattern and formyl med, right? Part of the DNA sequence of the bacteria. So these are all considered endocytic, which usually present on the surface of phagocytes. And normally when they recognize when they have this kind of receptor, they will be able to engulf the pathogen and digest them. So there is endocytic pattern recognition. That's why they are usually available on the surface of the pathogen. So including scavengers, receptors, opsonine. So for opsonine, the difference between the previous type of receptor is basically they are not recognizing the bacteria directly. But instead they recognize the intermediate intermediary protein, which is the coating protein. So still remember the function of opsonin to tag, right? To coat, right? We know before. So these are example of opsonin molecule. C3b is an opsonin. So they can bind to the surface of the pathogen. So therefore, phagocytes with the receptor, they can bind to the opsonin. Opsonin will bind to the opsonin. So they are not directly binding. They bind to the opsonin instead of the bacteria itself. Whereas there is a different type of recognition receptor, which is known as signaling pattern. So the difference with this receptor is usually when they bind to the bacteria, so they will cause significant changes on the immune cells, and they start to produce effect. For example, they produce cytokines, they produce enzyme, so they produce something. That's why it's known as signaling pattern recognition receptor. And these are the example of the receptor could be found. It's toll like receptor, CD40 receptor. So all of these TLR are basically toll like receptor. And the signaling pattern recognition receptor usually could be on the surface of the immune cell as well as in the membrane of the endosome and the cytoplasm. Salam. Wait, guys. Yep. Sorry guys for the interruption. Are you still with me?
Yes, doctor.
Yes, Doctor.
Okay, okay, just now was signaling receptor, right? So what's the difference between the previous receptor, guys? Signaling pattern recognition comparing to endocytic pattern recognition receptor. What's the difference? From the word itself, you can know. Here, the recognition of the receptor causing the cell to receive a signal instruction from the outside cell to the inside cells. So the difference is because of the distribution of the receptor itself. Before this, if it's endocytic, most of them are on the surface. But for signaling, they could be on the surface, could be membrane of the endosomes, could be in the cytoplasm inside the cells, right? So it could be on the surface, could be inside the cells.